Hey guys, welcome back to our last lecture here for the slightly saddening chapter 14. It's been a long talk of diseases and things of that nature, but this is our last one and it's been interesting, I hope. So we're going to talk genetic screening today. A few of these are going to be kind of some hot button issues for a few of us, but it's going to be a good talk either way. So genetic screening, our understanding of human inheritance can provide prospective parents with information about the health of their future children. So the idea of knowing a little bit about your kiddo's genetics before your kiddo is born is what we're going to be talking about. You could argue that the more you can approach things, surgery, prescription drugs, hormone therapy, even controlling what they have in their diet can minimize, or in a few cases, can actually eliminate symptoms of a certain genetic disorder. So this knowledge can be a very good thing. Couples may choose to know if their future children face a high risk of inheriting a severe genetic disorder through this genetic screening and genetic testing, both on the parents and the brand new baby. Genetic analysis starts with your parent karyotypes, so literally getting just mom and dad's genotypes laid out on the table there in a picture called a karyotype. You can even look at their pedigrees. What traits did they have? What traits did their grandparents have? Their parents, their great grandparents, things like that. Just like the pedigrees we've done in class. And then finally, you can do genetic tests for known disorders that mom and dad might already have or might be carriers for and, uh, and not know it. And so all of these things might sound like, wow, that's too much. I don't know if I want all that knowledge and pressure on me, but it can provide some information for those who desire to pursue it. They use that information to predict the probability that they'll have a child with a particular genetic disorder. Most of these probabilities are very, very small, but some folks want to know this stuff. So, some disorders can be detected early enough to actually start these countermeasures before the symptoms develop. One example of this is a disease called PKU, or phenylketonuria. Now, most of your hospitals will screen newborns for mutations that cause PKU because it's one of those diseases that if we change their diet, we can help them a lot. PKU, this is right out of your book, is a mutation that affects an enzyme that converts phenylalanine into tyrosine. So when they have the disease without the enzyme, the body becomes deficient in tyrosine and phenylalanine accumulates to high levels. The imbalance inhibits protein synthesis in the brain, which results in severe neurological symptoms. So basically, this is going to cause slow brain damage, having this disorder or disease, and that's a guarantee. But if we know that and we can change this kid's diet and keep them from eating foods that are high in phenylalanine, we can actually help them survive a lot longer and live very normal lives with really pretty much totally normal neurological function. And that's a big thing you can do with such a small change like diet. And so you'll see that on food labels. It'll say on the bottom of the food label, caution phenylketonurics, this food or drink is high in phenylalanine. And so that's the reason those warnings exist. So if you get this diagnosis prenatally before the kiddo is born, the embryo or the fetus is tested before birth to screen for the sex of the child or genetic problems of the child. That's what prenatal means, before birth. Okay, so prenatal diagnosis. There's lots of ways to take a look at a kiddo before the kiddo is born. The non-invasive technique that I'm certain you guys have seen in movies, TV shows, is called obstetric sonography, which is where they take that little blue goop on mom's belly and they take a little, white-handled machine and they rub it over mom's belly and it sends sound waves into the uterus, and those sound waves reflect back off the baby and give us a picture. Okay, and people are familiar with that. That's how you get your first, we generally call it an ultrasound. You get your first ultrasound picture of the new baby, and it looks like a little gummy bear or something like that. And so that's one option. Now it's going to give you, they're getting pretty good actually. They get some 4D ultrasounds that are pretty sharp, but it's going to give you a decent picture of major internal organs, even blood flow now, that sort of stuff. 5 fingers, 5 toes, unless you're polyploidy, and you know that makes sure the spinal column's inside the body the whole way, that kind of stuff. But you don't get a real great high definition picture like you can get with a few other options. So if they suspect there's a problem based on the sonography work, the doctor might push for or request some invasive procedures. So not just outside of the body, an invasive procedure where you need to go in and get tissue, or you need to get blood, or you need to take a picture from the inside, that kind of stuff. And those options are, I hope I'm saying this right, fetoscopy, scopy, you know, like microscope, you know, to get an image of or to look, and feto is fetus, an amniocentesis, or chorionic villus sampling. And so fetoscopy is to pulse sound waves just like you do with the ultrasound, but they're pulsed from inside of the mother's uterus. And so what you're going to do is actually take a picture of that kid right next to the kid, and you are going to get an incredible picture out of that. And so you can try to look for genetic disorders with that. An amniocentesis is one that you may be asked about if parents want to pursue that as an option, and that is where they actually go in and draw out with a needle and syringe a little bit of amniotic fluid, and that fluid contains cells from the new baby, and they can check the genetics of those cells that they drew out of that fluid. So that's a little more invasive. And then there's chorionic villus sampling, CVS, and that's where they go and take a few cells from the chorion or the chorion, and that is a little membrane that surrounds the placenta. I'll show you a picture of that in just a little bit, and they test the genetics of those cells. But again, you got to get, you know, the needle, the syringe, into mom's uterus, right next to the baby. It makes people kind of nervous. Sometimes that chorionic villus sampling can disrupt that placenta a little bit, and you might get some underdeveloped toes or fingers, or even missing toes and fingers in the new baby. You're only going to see that, I think your book, 0.3% of newborns. It's not a huge number by any means, but some parents wouldn't want that risk. Also, fetoscopy, you know, getting that image right next to the kiddo in utero, is going to raise that miscarriage rate. It's going to boost the miscarriage rate by 2 to 10%. So, I mean, that's something everybody's got to kind of make a choice of for themselves. I'm not going to sit here and tell you what to do or what not to do, but you know, the more you know, the more informed your decision's going to be. So I guess you know about these, you know the information they can give you, and you know the risks that come with them. So I mean, you're sitting in an okay place to make an informed decision. If you have questions about them, ask me in class. I don't claim to be an expert, but I'm pretty sure we could find one if you want to. I'm not worried about doing that. We can sure chase down somebody who knows more about this than I do, and that doesn't bother me at all. This is the conventional ultrasound that is, you know, the blue gel on top of mom's belly that they're taking the picture of. They're quite a bit better than this nowadays. This is okay. I mean, it's kind of cool. You can see the kid's skull in there, which is neat. But they can get really, really good pictures now with these. The 4D ultrasound, which again is very, very good, and you can get a lot of detail. And this is that fetoscopy here where you're way, way close to that kid taking the picture. And so here you can see the shape of the face. Here, I mean, you can see the fine hairs on this kid's skin. You're so close. You can see fingernails, ends of fingers, I mean, blood vessels running under the skin. Like, this picture is pretty cool. So there you go. That's 3 different pictures of a human fetus, and the different methods of taking the picture give you a very different picture. Here's a quite a bit earlier on baby, and you can tell that from the eye, from the very short fingers and toes, and of course the relatively small size. Also the head is very, very, very large. And this picture is mostly here to show you the chorion. Okay, this is the kiddo. Kiddo has the umbilical cord, which attaches to the placenta. The placenta is basically a blood. It's a big bed of capillaries that meshes in with the capillaries of the uterine wall in mom, and gives the kid nutrition, and also removes waste through mom's bloodstream. So the kid makes this, not mom. This is from baby. And so all around this placenta, this edge right here, that's called the chorion. And in that chorionic villus sampling, these little guys right here, those are the cells they're shooting for to get a little information about that kid's DNA. Amniotic sac is filled with amniotic fluid. So in the amniocentesis, they have to stick a needle down here into the amniotic sac and draw out fluid from in here. So you can get a lot of information. The risk, that's up to you, of course. So what about pre-implantation? So all the stuff we've looked at is when there's already a baby. Implantation refers to the new zygote or blastula or morula or whatever it's called at that point, hooking on to the endometrium of mom's uterus, the innermost layer, and attaching and starting to form that uterus, or that uterus, goodness gracious, I'm sorry, that placenta, starting to form that placenta. Before new kiddo hooks on, that's called pre-implantation, where you've got a fertilized egg, so a zygote, new human, embryo, pick the term you like, tiny baby, whatever you prefer, okay? That's pre-implantations, before it's implanted on the uterus. And so some of these couples who have a high risk of having a child with a genetic disorder might choose in vitro fertilization for pre-implantation diagnosis. You might have heard this shortened up to test tube babies, right? In vivo is in life. In vitro is not in a living thing. So egg collected from mom, sperm collected from dad, make the zygote in, I don't know if it's truly a test tube, probably some sort of small shallow dish, make the kiddo in the lab. Kiddo starts to divide, implanted into mom. That's the idea there. So the undifferentiated cell is removed from that early embryo because as soon as sperm fuses with egg, it's this really big cell, and it's going to go through mitosis and divide into 2 cells and divide into 4 and divide into 8. And at that time, those cells are undifferentiated. They have all the information needed to make a brand new human, but none of them are brain cells or muscle cells or skin cells. They're not there yet. They're undifferentiated. They're a blank slate. And so what they can do is they can actually take one of those cells away. And to our knowledge, it's fine. The cell that they just keep dividing and becoming a cluster of cells and a cluster of cells, and they just keep dividing. And, to our knowledge, they don't even know that one cell is gone. So we can take that one undifferentiated cell away, and we can examine all of its genes prior to implantation. And if the embryo has no detectable genetic defects, it's inserted into the mother's uterus, assuming that it responds well to that and successfully implants, she would be pregnant with that new baby. If it contains the genetic defects, that would be up to the parents. My assumption, I don't know what would happen with that kiddo. It might be implanted anyway. If they want to go ahead and have it, it more than likely might be discarded. So that, of course, would strike some ethical and moral kind of hot button issues for a lot of us. But for right now, I'm just showing you what's going on. If you want to talk about what it means, right and wrong, we can certainly do that in class. But for a flipped class video, we don't want to do that just yet. So this is an embryo after 3 mitotic divisions. So undifferentiated cell, undifferentiated cell, undifferentiated cell, and it's just a cluster of undifferentiated cells. So that's a point where you could pull one cell out right here, just pull them out of there, test that nucleus, see what's going on in there for genes that might carry genetic defects or genes that will carry genetic defects, and this little cluster of cells would continue on dividing without that one cell, and to our knowledge, it would be just fine. So a couple points to ponder, and then that's going to wrap up the chapter. First question, how do sex chromosomes actually determine the sex of an individual? Second question, what are some of the characteristics that are governed by genes on the X chromosome and on the Y chromosome? And then finally, are any human traits Y-linked? So only found in males. Those are some questions to kind of chew over for a little bit. And with that, we are done with chapter 14. So write down your questions, and we'll visit about it in class. It'll be a good time. We'll talk about it. So thanks for your time.